Log on, tune in, find out. Another good idea from Cambridge.
Invitation here. I don't dare, I'm afraid, show any equations in such august company, so I hope you'll allow me to focus just on some pictures instead, and also a little bit more of an industrial and commercial perspective on what's going on in imaging for drug development at the moment. So I'm talking about Alzheimer's disease as a particular focus, and that's because I think the person originally invited to give this talk is a specialist in Alzheimer's disease, and I was going to kind of try and step into his shoes if I can. But I think some of what I say is also quite general. So I thought I'd start by telling those of you who don't know what IXICO or who IXICO is. So we're a medical imaging company. We are 6 or 7 years old. We were founded with sort of know-how and technology that came out of some places in London. You've probably not heard of them here in Cambridge. And what we do is we provide software for clinical trials and research studies. We actually help run those trials, and we also provide what's called a full service imaging CRO capability. So pharmaceutical companies in particular and some academics outsource to us the job of running all the imaging in their clinical trials. So we provide image management technology, archive and management. We do clinical trial image management for safety and efficacy, and we use biomarkers. And we do biomarkers both in late phase studies with biomarkers that are very mature, and we do work on more novel biomarkers moving towards a qualification of those. And so our vision as a company has been to bring the very best possible imaging technology and know-how to the R&D community and to the pharmaceutical industry, and therefore to help bring new treatments to market quicker. So just a word about medical imaging. So of course there's a huge diversity of medical imaging in vivo at organ level here, and obviously we're hearing some very impressive work on new modalities, sort of photoacoustic, which is now looking like it is becoming able to, close to becoming able to image organs. But the primary modalities are really CT, MRI, ultrasound, X-ray, PET, and SPECT. In healthcare, of course, the scanners and the workstations are dominated by the major manufacturers of light bulbs, and the contrast agents people inject tend to be made by the specialist diagnostic pharmaceutical companies like GE Healthcare and Bracco. And research is aimed at two things really: research with imaging, understanding normal development and natural history of disease, and also testing the safety and efficacy of new drugs. And it's that last point that I want to particularly focus on. But as you'll see, there's a very close relationship between that and what goes on in healthcare, what will go on in healthcare. So looking at R&D, the most important point here is that for research application in medical imaging, almost, not always, but almost invariably, we want things to be quantitative. And the reason we want things to be quantitative, we want to feed it into some statistical analysis in order to work out what's going on. Does a drug work, or what happens in the population in terms of natural history? Whereas in healthcare, of course, we're more often looking at qualitative analysis, and it's in the first of those, in the quantitative, that the mathematical challenges lie. Qualitative analysis involves looking at the images with a radiologist who uses their experience and their expertise to try and identify are there differences from normality there, and how does that impact on a differential diagnosis. In the quantitative world, we need to do something which gives us numbers. And one of the big problems with images is they're inherently very highly dimensional, and we need to, for statistical analysis, have a very, very small number of numbers that we can use for decision making. Particularly, in general, statisticians don't like a situation where you've got many, many more numbers in your voxels than you have patients in your study. So you've got to get summary statistics which you can do your calculations on. Now, most imaging research is done at a single centre, and you carefully optimise the way you collect the data and analyse it. But in the sort of clinical trial I want to talk about, I want to focus on, we're talking about really huge experiments. We're talking about experiments that might have thousands of patients required in order to answer the question. Thousands of patients typically for most therapies it's tough for any one hospital to recruit more than one patient per month, often a good deal less than one patient per month. So if you want thousands of patients and you want to actually enroll people into your study in less than a year, you're typically talking about 100 or more sites. So it's a huge issue about how you do a scientific experiment with imaging over 100 or more sites, particularly when your competitors, other pharmaceutical companies, are also trying to test similar drugs on similar numbers of patients on similar sites. And I think it's worth really noting a very important problem here. Drug development has been a very unsuccessful endeavour in the last decade, and that the big pharmaceutical companies are kind of at the moment shedding staff in the research area like trees shed their leaves in autumn because of a real problem in getting drugs all the way through to approval. Billions and billions are spent every year by big pharma companies, and I think we've got somebody here, Bill from Pfizer, and I don't know how much they spend a year, 8 billion a year or something, and over the last 5 years, how many small molecules have come out and got approved?
In the last 5.
Is it that many? Okay. But it's still, the average number is very, very small. Really, really low R&D productivity. Billions and billions spent for every one molecule that gets approved. Now, having said that, imaging is potentially able to help with that if it can be used right. And giving ways of getting metrics of drug safety and efficacy. And I'll come back to some real case studies of this in a few moments. But one thing to look at here is that over the sort of in the 80s and 90s, the cost of developing drugs just rocketed up, as you can see in this graph here. Now, it has, depending on how you look at this, some of those numbers have begun to plateau now because pharma has started really reducing their costs. But that's clearly an unsustainable situation. So imaging can assist in drug development through early phase decision making. So that's before you do these really, really big trials, trying to answer questions on small numbers. And also providing evidence of disease modification. So showing that a drug doesn't just help give you relief from symptoms, but actually changes the course of the disease. There's also a very important role in translating into healthcare. You've probably heard the buzzword of companion diagnostics. People also talk about personalised healthcare, the idea that the way you give a drug to a person is very customised to their particular genotype and phenotype. So the way that imaging is used in drug development, there's also an interesting parallel there to how it might be used in what I'm calling the diagnostic algorithm, the way people are selected for treatment later on. So you want to select the patient you'll benefit, and you want to avoid treating patients who might suffer side effects from their benefit. So regulators are very concerned about both those things. They don't want to have people who are sick not getting a drug they need, and they don't want people who don't have that disease but to get a drug which might have some side effects when it could have no benefit. And so there's that need to get that balance right, and that of course in doing imaging to test drugs, there's a question about how imaging then gets used in the diagnostic algorithm later. And I'll come back to this point a little bit after I've given some examples. And those examples are in the area of Alzheimer's disease. So Alzheimer's disease is a very important topic to all of us. I think it's probably fair to say that a third of us in this room are likely to get Alzheimer's disease in our lifetime. And the reason it's higher than average in this group is that the incidence of Alzheimer's disease is higher than average in people of above average mathematical ability. But here we see 3 pictures of Alzheimer's disease. We've got a molecular picture here. So the amyloid precursor protein here, and then these enzymes which chop it up, the gamma secretase and beta secretase enzymes, the amyloid fragments which then aggregate into these plaques, and these plaques are what you see post-mortem in people who have died of Alzheimer's disease. But it's also true to say that probably 40% of 70-year-olds without Alzheimer's disease also have these plaques in their brains. So these plaques come along before you actually get clinical Alzheimer's disease. And here's a microscopic picture. So here's what happens if you take a brain and you use a dye to stain it, to then find the amyloid and look at the amyloid. And then here's a structural picture. So here's a brain of a fairly normal subject in the coronal section, grey matter and the white matter, language regions around here and memory here. So although it's a structural scans do tell you things about function, because different bits of the brain, structural pieces, are associated with certain functions. And if we see what happens in dementia, we see bits of the brain shrinking, the memory bits, and language centre slightly later go. The bit that's preserved for longest in Alzheimer's disease is visual cortex, unless you've got posterior cerebral atrophy when your eyes go early in the disease. So looking first at the structural imaging, what is it that we might try and do? So the word for kind of the brain shrinkage we see is atrophy. And that Alzheimer's disease is characterised by a rate of atrophy that increases slowly over time, and it's about 3 or 4 times that of elderly normal controls. Familial studies, so that's the very unusual sorts of Alzheimer's disease where people have a genetic predisposition. You saw the amyloid precursor protein on my previous molecular diagram. Some people have an APP mutation which gives them a familial form of Alzheimer's disease. You can obviously genetically screen those people and then and do long-term studies, and they develop Alzheimer's disease, and you can use that as a cohort to see, well, what happens years and years before they get the disease, and that you see brain loss well before diagnostic criteria are met, and that's helped validate these methods for natural history studies. So what we can do is we can say that over the course of years here, these are days, but many thousands of them, over the course of years we see for people who are developing Alzheimer's disease, their brain volume as a percentage of the whole brain, total intracranial volume, whole head volume, it gradually accelerates, and that you don't meet your diagnostic criteria down here, and clearly there's a lot happening. So one of the points about this diagram is that there's a lot happening years before you get Alzheimer's disease, and so maybe you want to start treating people 2-3 years before their clinical symptoms. I'll come back to the point in a minute, but just bear in mind that there's a lot happening before your doctor would say you've got Alzheimer's disease. Now just see some pictures. Here's a normal control, and we can just see images a year apart, and you can see very little change. These are images courtesy of Nick Fox at UCL. If we look at somebody who's got a cognitive deficit but not yet clinical Alzheimer's disease, this is somebody, it's called a condition which is sometimes called amnesiac mild cognitive impairment, or MCI here for short. And here you can see a change in the brain. So whereas in the first image we saw very little change, here we can see quite distinctive change, most obvious perhaps around this part of the brain, but also in the sulci here, hippocampus, around there. So we're seeing that pattern of structural change we saw in that cartoon earlier over a year. And here's somebody with full-blown Alzheimer's disease, and we can see just in this particular case 6 months, we can see an even more obvious set of changes going on. Now these are patients kind of at the upper level of that progression, so it's not so obvious in everybody, but these are good scans for illustrative purposes. So that's about what we can see in the structural scans, and a big challenge that people have worked over the years has been how we quantify that. And I thought there's I was actually talking to somebody in a pharmaceutical company earlier who was talking about one of his colleagues who's giving up on drug development and going into finance, and that same thing happened actually in the person who developed the first algorithms to quantify that atrophy, a guy called Pete Freeborough. He developed the boundary shift integral method of quantifying that. He then gave up academia. He went to work in the banking sector, and in 2007-8 he was trading in synthetic CDOs, and he led directly to the collapse of the banking system, which I think demonstrates the risks of people leaving R&D and going into banking. We need the creative algorithm writers to stay in the science world, I think, and keep out of banking. But the next little piece to look at, a completely different way of imaging, is the microscopic picture here. Now, do you remember I said that these are stains that you can apply to tissue? So some people in Pittsburgh came up with an idea of saying, well, let us use a sort of adapt that, trace that molecule that you use as a label in your stains, and try and actually inject it into the patient and let it go into the brain, and then we can actually, if you like, stain the whole brain. This is radio labeled with carbon-11, which is a positron emitter, and so you can then stick something in a PET scanner and you get an image here, and that here you can see that you might have an Alzheimer's disease patient with a lot of uptake of this tracer in their brain, and a normal control with much less, and the mild cognitive impairment, people who are perhaps between the two, though in fact it's not as simple as that. Some of the MCIs will tend to be positive and some will be negative. Those that are positive are much more likely to develop Alzheimer's disease than those who are negative, who tend to be less likely to. At least when I say less likely over, say, a 2 or 3 year period. And here is a picture of the website of a company that develops these drugs, which of course, because it's the company's website, it looks that much clearer, the elderly control and the Alzheimer's subjects. The thing to really watch here is that 40% of people who are around 75, 80 who probably have this kind of signal in their brain, even though they've not got Alzheimer's disease. They've got that molecule in the brain, but they haven't yet had the symptoms. So this is a slightly simplified story, but then this company here, Avid, the company that made this, had a particular goal in mind when they were developing all these slides, which was to be bought. And they were bought by Lilly for around about $1 billion, I think, including an earn-out. So Lilly now bought this tracer from Avid, and now Lilly has a tracer for Alzheimer's disease and is trying to get that for at least approved for diagnostic use in the US this year. They were hoping, though it may be next. The reason that they bought it, incidentally, was because they were having so little luck in getting any drugs through their pipeline that they thought, well, let's go buy something. We really need an approval, FDA approval in 2011. And so they bought this, went to the FDA in January. FDA weren't quite happy with the way that it was analysed, and so it bounced back. So it's probably not going to be approved till 2012. So I guess the CEO won't get such a big bonus as he was hoping. But then, well, on the subject of Lilly, one reason Lilly wanted to buy this was that they had more bad news just earlier in the year. So this is back in almost exactly a year ago, 17th of August last year, so semagacistat was a gamma secretase inhibitor. So do you remember that picture I showed earlier where you had the kind of APP and being chopped by enzymes? So gamma secretase is one of those enzymes, and the idea is if you can inhibit that, you can then stop the chopping up of the amyloid, and therefore you can stop the amyloid cascade. You can slow it down so that you don't get the fragments which then aggregate and so forth. So they did this big study, and this was one of the most serious programs that we've yet seen in the world of Alzheimer's disease treatments. It was many hundreds of millions was spent on this program, and it is true to say that it's the very first study in Alzheimer's disease that has achieved statistical significance in the primary endpoint. Yeah? So the primary endpoint was improved cognition, and they got statistical significance. Unfortunately, it went the wrong way. So it was worsening of clinical measures of cognition and activities of daily living. That's not what they wanted. So I remember having a chat to one of the statisticians at Lilly on the 18th of August, and she was saying they spent quite some time making sure they hadn't mislabeled the two groups, because obviously they were quite surprised when they unblinded to find that the effect went the wrong way. And this was actually done in interim analysis, not at the end of the study. And so they terminated the study early, as it says up here. But this just illustrates a huge problem. And the huge problem is that you can take a study to the level of doing thousands of patients and then find not only does it not work, it makes people worse. And not only did it worsen their cognitive status, it also gave 5% of them basal cell carcinoma. Now that you can treat, and everybody was treated, but you have to resect it with a margin, so it's not a really nice treatment. So, if you've got, all the whole population of Alzheimer's disease, yeah, then, basically train to be a surgeon who takes out basal cell carcinoma. You have a job for life if these drugs got approved. But the key challenge here is how on earth are we going to deal with this problem? How are we going to deal with the problem of getting a drug which looks great in every measure you can do early on, and then it falls over because of lack of efficacy. If it were good enough, yeah, then the side effects would have been manageable. But because people will accept side effects for treating Alzheimer's disease because it's such a hideous disease. But it just, to get to this stage, to largely finish the trial, doesn't work. So I want to look at another molecule, a molecule being developed jointly by two companies called Pfizer and J&J, so two huge companies. Now, it didn't used to be being developed by Pfizer and J&J. It was being developed by a company called Wyeth and Elan. And Pfizer bought Wyeth and Johnson & Johnson bought Elan. And I'll tell you why Johnson & Johnson bought at least that bit of Elan. So this was a study on a very small number of subjects, 26 or something, who were treated with this drug. And the drug is called bapineuzumab. So it's a monoclonal antibody which targets the N-terminal of the alpha amyloid beta 142 segment. And what they have done here is you're looking at screening patients in week 78. So that's the very first scan. That's the week 78 scan. And this is the looking at the treatment of those who are treated versus those who are on the placebo, and a couple of examples. And what you see when you start looking at the graph overall is that as you go over the 78 weeks of the study, you see that in the bapineuzumab treated patients, you see that the amount of amyloid in the brain is decreasing in the bapineuzumab group, and it's increasing in the placebo-controlled group. So that was a very, very small study, that is one of the main reasons that Janssen, or J&J, specifically Janssen, then bought Elan for about $1 billion. So imaging has real commercial value in this world. But I mean, the reason that's interesting is it's so difficult to get evidence that a drug works directly, that just evidence that you hit the target, that you can take a drug which is designed to reduce the amount of amyloid in the brain and actually demonstrate that in the treatment population in a non-invasive way is a huge, huge benefit. So, what people are excitedly waiting for now is what is happening in the phase 3 of this data, where they haven't just got 26 patients, they've got several hundred. There's thousands altogether being studied, but a few hundred of those are having the amyloid imaging. Will we get similar effect results again? So it's a fascinating biomarker experiment. So getting back to the very basics, so I've been trying here to give you a kind of a taste of the application. In the last 5 minutes before tea, I'm going to talk a little bit more about how we try and do all this. So in a clinical trial setting, you get data from sites, and there may be hundreds of them. You have to do all sorts of basic kind of stuff and a little bit of clever analysis down here. And you have to make sure that you then send the results to the pharma company, the sponsor, in a way which is fully regulatory compliant. You've got to obey all the laws about doing research in clinical trials. And so those are huge constraints on what you do. And that when you're explaining things to regulators, you've really got to remember that they glaze over when you talk about conformal mapping and Riccian geometry. They have real problems with that sort of language. So if we are looking at some of the challenges, ideally we could answer questions on small numbers of subjects at early stages, but we still need to be able to do these big studies, imaging in these big studies. And for imaging in these big studies, what really matters is we can actually recruit patients. That's by far the most important thing. Much more important that we can recruit patients than that we've got a really good imaging centre at those sites. And so we have to deal with sites that are not necessarily very experienced or very expert. So just talking about again the Alzheimer's example, we can use Alzheimer's disease. So we can use both MRI and PET to help us try and assess some efficacy and to assess some therapy. I haven't spoken about that very much, but we can look at the images and we can look for signs of side effects in the brain resulting from that taking the amyloid out of the brain. You can see side effects in the brain sometimes before you get the patient is sick. And you can also use imaging to try and say, well, we now want to treat earlier. So rather than just say we'll treat everybody who has got clinical Alzheimer's disease, maybe that's too late. Maybe we need to treat people who haven't got clinical Alzheimer's disease, but they would get it in 2 or 3 years' time. And so people are now saying, well, let's not treat people who've got mild to moderate Alzheimer's disease, let's treat people who've got preclinical Alzheimer's disease, sometimes called prodromal Alzheimer's disease. And for that, you need not only to have some cognitive deficit that you can measure with a memory test, but also you need to have some biomarker that you can use to help identify people who are likely to convert. And therefore, you can do a study with a manageable number of patients because enough of them will convert over, say, 2 or 3 years, that you can do a 2 or 3 year study. Well, when I say 2 or 3 year studies, each patient is on for 2 or 3 years. Yeah? A 2-year study is likely to take 5 to 6 years overall, including all the recruitment. And a 3-year study is likely to take 7 years. And remember, the patterns only last 20 years. So these are huge undertakings to actually do these studies. So there's loads of endpoints that you can use, and I'm not going to go into the details of all these, but some of them are much more appropriate at early phase and at late phase. And so I'm going to sort of just nip on just a couple of thoughts about mathematical challenges here. So it has to be said that an awful lot of image analysis methods have pushed the frontiers, AD methods have pushed the frontiers of image analysis. And that there's a lot of maths that underlies these. I'm not going to talk about this in detail, but I think there are some very important challenges that remain, which is particularly how we deal with measures that really work within this global environment and the regulatory compliant environment, because clever maths isn't enough in itself. We have to be sensitive to what we want to measure and as insensitive as possible to things that we don't want to measure, like instrument and site variability. We also have to properly capture the variations we have in different populations in the globe, which we probably aren't doing very well at the moment. And we mustn't forget the regulatory environments where we have to have things that are transparent and easy to explain. So just very quickly, amyloid analysis. Here's an example of an image analysis workflow with various registrations and normalization and segmentation of grey matter to enable us to work out quantitatively whether or not we are seeing a change in the amyloid load in the brain, as I gave as an example in that earlier study. Wrong button. And then just a quick, another sort of anecdote looking at the latest kind of approach that's reached maturity for clinical trials in delineating regions of the brain is these multi-atlas methods. So here you have an atlas, and your goal is to label an unseen image. So there's your unseen data, but the concept here is that you actually take a whole load of different atlases because one is not enough. We had that kind of discussion earlier about what is the right mapping. And the answer is, of course, that there is the mathematical mapping that can go from one atlas to any subject can't be described in simple kind of diffeomorphic terms. So what you need is lots of different atlases which represent variability in the population, and then use all those atlases and then have some clever way of determining which is the best match on the patient, so a decision fusion approach. So this is a sort of method that people have now been using, and here's just an example of it here's a target image, which is a patient who has got quite a lot of atrophy, big ventricles, big black bits in the brain. And here's the 2 different views, an axial and a sagittal section. And here we have an atlas. So here is a manually drawn round hippocampus, and you'll notice here somebody who's got much less atrophy, very little atrophy and very little CSF visible in the sulci, small ventricles. And of course you use that atlas to try and label this patient, and we just see here that it just does not work. We deform the atlas and it just does not match the target image at all well, because if you've got so much CSF here and there's no CSF there, you can't literally tear, you can't, with these algorithms that use for registration, whether they work by kind of free-form deformations or fluid flow, you can't tear the transformation and introduce a big piece of CSF which wasn't there before. And so that kind of conceptually is why you need the multi-atlas method. And here you can see another atlas, and this atlas, of course, then works very much better. So it's atlas 2 that we need to use in this particular case, and we have to have a way of automatically working out which is best. So those mathematical challenges are important, but I want to get back to some of the practical, because it doesn't matter how good our algorithms are, unless we can get good longitudinal data across many, many centres in these late phase trials, we'll never get results that are usable. We have to do things quite quickly. Just the logistics of getting the data from all the places it's collected to somewhere to do the analysis to check it's been done right is a huge challenge. And if the data piles up for years before you analyse it, you find that a lot of it can't be read. You have to have an analysis method that is acceptable to regulators, and you have to deal with the kind of complex data in a way which deals properly with security, logistics and regulatory compliance for both confidentiality and for what's called GCP, good clinical practice. So there is this great role for imaging in R&D. Getting the good data is a huge challenge. The mathematics of the analysis remains something that we need to improve, but it's still the case that unless we get good data in, we're never going to be able to get good results. And that but what we are going to be seeing, I think, is increasingly the sort of things people are using to try and find patients for clinical trials and measure efficacy are going to start being used in the diagnostic space later, because it's very likely that Alzheimer's disease is an example of a disease where you're not going to have a one-size-fits-all therapy. We're going to need to stratify the population in the clinical trials and say which patient does it work on, and then an organisation like NICE might say we will only pay for this drug for this particular subgroup of patients. And to identify that subgroup, you probably won't just have clinical signs, you'll have to use some sort of quantitative imaging biomarker. So there's a lot of very interesting things that are going to happen, and there's a very compelling link, I think, between what's going on in the clinical trial space and in the healthcare space. At that point, I will finish, just I think 2 minutes late, so that's okay.
I think I'll leave questions for T and for the panel discussion, if that's all right. T is served immediately outside.